All right, welcome back, everybody, to the deep dive. We're diving into the world of viruses today, more specifically, viral genomics. It's amazing to think about how these tiny little things can pack so much information in their genomes and just exist in so many different ways.
Yeah, and it's fascinating how much diversity there is in such tiny genomes.
So for this deep dive, you've brought an awesome chapter summary on viral genomics and diversity. We're going to try to give our listeners a solid understanding of, how viruses store their genetic information, replicate, and how we even go about classifying them.
Absolutely, it's going to be quite the exploration.
So where should we even begin with this? I guess starting with the basics, like how do their genomes actually, compare to what we see in cellular life?
Well, the first striking thing is that unlike cellular life, viruses aren't limited to DNA as their genetic material.
Oh, wow.
They can use either DNA or RNA. And to add another layer of complexity, both DNA and RNA genomes can come in single-stranded or double-stranded forms.
So it's not just the four bases, it's the structure of the molecule as well.
Exactly. And in general, these genomes are much smaller than those of cellular
You
organisms.
streamlined, right.
Exactly. Efficiency is key.
Now, the summary mentions that viral messenger RNA, mRNA, is always considered to be in the plus configuration. What does that mean exactly?
Essentially, think of the plus strand as the coding strand. It's the sequence that can be directly read by the host cell to produce proteins. You can almost think of it like an instruction manual.
Okay,
The minus strand is its complement, like a photographic negative.
gotcha.
So if a virus has an RNA genome that's already in the plus configuration, it can act immediately as mRNA once inside the host cell, ready to make proteins.
Oh, that's pretty efficient.
Right.
Okay, that makes sense. The chapter also introduces the Baltimore classification scheme, which sounds pretty important for understanding viruses.
It is, absolutely. It's like our Rosetta Stone for viruses. This system groups viruses into 7 classes based on their genome type and how they produce mRNA. There are 3 DNA-based classes and 4 RNA-based classes. And just by knowing a virus's Baltimore class, you can get a pretty good idea of how it replicates and expresses its genes.
So it's a quick way to kind of categorize their overall strategy.
Always.
Now, one thing that stood out to me was this idea that RNA viruses need RNA-dependent RNA polymerase, something that DNA viruses don't require. Why is that such a big difference?
That's a fundamental difference because our cells, they can transcribe DNA into RNA. That's how our genes are expressed.
Right.
But our cells don't have the enzymes to make RNA from an RNA template.
Okay, so they have to bring their own equipment.
Exactly. RNA viruses have to bring their own or encode it in their genome. This RNA-dependent RNA polymerase, or RNA replicase as it's often called, is essential for them to replicate their genomes and make more mRNA.
It's like they come with their own specialized toolkit.
Absolutely.
So how do scientists actually go about classifying and naming these viruses with all this incredible variation? The chapter mentions a polyphasic approach and the ICTV.
So viral taxonomy is a complex field. And the ICTV, the International Committee on Taxonomy of Viruses, is the governing body for it. They use this polyphasic approach, meaning they consider multiple factors.
Oh, okay.
So things like what kind of organisms the virus infects, its structure, its replication cycle, and increasingly important, the actual sequence of its genome.
So it's not just how it looks, it's what's under the hood, so to speak.
Exactly. And they also look at evolutionary relationships, the phylogeny of the virus is like building a family tree.
Right. So it's this hierarchical classification system we see for plants and animals too, right? Orders, families, the whole deal.
Precisely. It helps us organize this incredible diversity.
Hmm.
However, figuring out the evolutionary history, that phylogeny, for viruses, can be quite tricky.
So what makes it so challenging to trace their evolution?
Well, unlike bacteria and another cellular life, viruses don't have a universal marker like ribosomal RNA that we can use to compare across all of them.
So it's harder to find those common ancestors, those links.
Exactly. Plus, viruses mutate much faster than cellular life, making it hard to trace their lineage over long periods. It's like trying to follow footprints in shifting sand.
I can see how that would be tough.
So we often have to rely on comparing specific proteins that are conserved within certain groups of viruses to construct their phylogenetic trees. And the sheer diversity, especially among viruses infecting archaea and eukaryotes, is so vast that many haven't even been classified into orders yet.
Wow, so there's still so much we're discovering.
Absolutely. It's an exciting field.
It sounds like it. Now, the chapter makes a general observation that prokaryotes and eukaryotes tend to be infected by different types of DNA viruses.
Yes, that's a general trend. Prokaryotes, like bacteria and archaea, are mostly infected by double-stranded DNA viruses, whereas eukaryotes can be infected by all 4 types of genomic configurations we talked about.
Interesting. So let's get into some specific examples of DNA viruses. We'll start with single-stranded DNA bacteriophages like FX174 and M13. FX174 sounds really interesting with its overlapping genes.
FX174 is a great example of genomic efficiency. It has this tiny circular single-stranded DNA genome, and what's amazing is that some of its genes actually overlap.
Overlap, what does that mean?
It means a single stretch of DNA can code for more than one protein, usually by using different reading frames. It's like writing a secret message within a message.
So it's squeezing as much information as possible into a tiny space.
Exactly. Once inside a bacterium, this ssDNA is converted into a double-stranded form, called the replicative form.
Okay.
This serves as a template for both mRNA production and genome replication, which happens through a really cool process called rolling circle replication.
Rolling circle replication, that sounds kind of fun. What's that all about?
Imagine a circular piece of DNA and an enzyme nicks one of the strands, creating a free end. Then a DNA polymerase comes along and starts extending this end, using the intact circle as a template.
So it's just going around and around?
Yes, and as it goes around, it displaces the original strand, which is then used to make new complementary strands.
So it's like constantly unrolling and making copies.
Exactly. This can produce many copies of the genome linked together like a chain, and then they're cut into individual genomes. It's very efficient. In FX174, the A protein nicks the DNA and also joins the ends back together after replication, while the E protein helps the host cell burst open to release the new phages.
So it's not just replicating, it's orchestrating the whole escape plan.
Exactly.
Now, what about M13? It's also an ssDNA bacteriophage, but has a different life strategy.
Yeah, M13 is a filamentous phage, so it's long and thin. And like FX174, it has a single-stranded DNA genome that forms a replicative form. But the key difference is that it doesn't kill the host cell when it releases new phages.
Oh, so it's more of a long-term relationship.
Exactly. New phages are continuously extruded through the cell membrane, so it's a chronic infection.
Okay, so it's a steady stream of new phages instead of a burst. That's really interesting. Now, moving on to double-stranded DNA bacteriophages, we have T4, T7, and lambda. T4 is known for having this modified base, 5-hydroxymethylcytosine, in its DNA.
Right. T4 is a large, complex phage with a linear double-stranded DNA genome. This modified base protects its DNA from the host's restriction enzymes, which are like a bacterial immune system that chops up foreign DNA.
So it's a way to evade the host's defenses. Clever.
Very. Plus, its genome is circularly permuted and terminally redundant.
Can you unpack those terms a bit? Those sound pretty technical.
Sure. So imagine you have a bunch of T4 genomes all lined up. If you were to cut them all at the same spot, the order of the genes would be different in each one.
Oh, wow.
That's circular permutation. And terminal redundancy just means that the ends of each genome have repeated sequences. This happens because the DNA is packaged into the phage head from a long chain of linked genomes.
So it's like filling a box to the brim, and sometimes you get a bit of the next item in there, too.
Precisely. And T4 is really self-sufficient. It encodes its own DNA polymerase and even modifies the host's RNA polymerase so it can control when its genes are expressed, like a conductor leading an orchestra.
So it's a highly organized takeover of the host cell.
Absolutely.
Now, what about T7? It's also a dsDNA phage, but what makes it special?
T7 is another one with a linear genome and terminal repeats.
Okay, so similar to T4 in that regard.
Yeah, but T7 encodes its own RNA polymerase specifically for transcribing its late genes. It also has an anti-restriction protein, like T4, to protect its DNA. And its genome replication is bidirectional, meaning it starts at a specific point and goes in both directions.
So it's got its own specialized tools for replication and defense.
Exactly.
And then we have lambda phage, which is described as a temperate phage. That sounds really intriguing, like it has a choice in its lifestyle.
Lambda is fascinating. Its linear genome circularizes inside the host. And here's the cool part. It can choose between 2 pathways.
2 pathways.
The lytic pathway, where it replicates like crazy, makes new phages, and bursts the host cell.
Classic viral behavior.
Right. But it can also go lysogenic. In this pathway, it integrates its genome into the host's chromosome, becoming a prophage.
So it basically goes undercover, hiding out in the host's DNA.
Exactly. It's a silent passenger replicating along with the host.
So what determines which path it takes?
That's controlled by a delicate balance of 2 repressor proteins, CI and CRO. These proteins regulate which viral genes are expressed, essentially acting as a switch between lysis and lysogeny.
So it's like a molecular decision-making process.
Fascinating. Now let's move on to DNA viruses that infect archaea. The chapter mentions they're mostly dsDNA and often have really unusual shapes.
Yeah, archaeal viruses are a trip. Most of them are double-stranded DNA, often circular, but their morphology is all over the
What
place.
do you mean?
We've got some head and tail viruses, but also spindle-shaped ones, rod-shaped, and even pleomorphic viruses that can change shape. It's a wild world. Some even undergo post-release development, like APV, which develops tail-like structures after being released from the host. So they're still changing and evolving even after leaving the host. That's wild.
Totally. And while many rely on the host's RNA polymerase for transcription, they can also encode their own transcription factors to regulate gene expression. And their release mechanisms can be pretty unique too, like sulfolobus viruses that form pyramid-like structures to escape the host. Plus, we've also found some single-stranded DNA archaeal viruses, just to add to the diversity.
It seems like archaea and their viruses are a whole different ballgame compared to what we see in bacteria.
Absolutely. Given the extreme environments archaea often live in, like super hot springs and acidic pools, it makes sense that their viruses have evolved these unique strategies.
Okay, shifting gears to uniquely replicating DNA animal viruses, the chapter highlights poxviruses and adenoviruses. Poxviruses are interesting because they replicate entirely in the cytoplasm, which is unusual for DNA viruses.
Yeah, most DNA viruses need the host's nucleus for replication, but poxviruses are like, nah, we got this.
They're independent.
Totally. They're large viruses with linear dsDNA genomes, and they encode all the machinery they need for replication and gene expression in the cytoplasm. It's like they bring their own portable lab.
They come fully equipped. And what about adenoviruses? They have this unique thing with inverted terminal repeats and a terminal protein.
Adenoviruses also have linear dsDNA genomes, and at each end they have these inverted terminal repeats, which are identical sequences but flipped.
Okay.
And there's a protein attached to the ends called the terminal protein.
What does that do?
This protein acts as a primer for DNA replication. So when the genome enters the nucleus, these repeats allow it to temporarily form a circle.
Okay.
Then replication happens only on the leading strand, using the terminal protein as a starting point. It's a really clever way to avoid the complexity of lagging strand synthesis, which is usually needed for linear DNA replication.
So it's a streamlined way to copy their DNA. Very cool. Now, the last group of DNA viruses we'll discuss are the DNA tumor viruses, specifically polyomaviruses and herpes viruses. These obviously raise concerns because of their link to cancer.
Absolutely. Polyomaviruses like SV40 have small circular dsDNA genomes and use the host's machinery for replication. The problem arises when they infect non-permissive cells where they can't complete their normal cycle.
So the replication process gets disrupted.
Right. In these cases, the viral DNA can integrate into the host's genome, and some of the viral genes can mess up the cell's growth control mechanisms. This can lead to uncontrolled cell division, which is essentially what cancer is.
So it's like the virus accidentally flips a switch that turns on uncontrolled growth.
In a way, yes. And herpes viruses are also linked to cancer, but they have a different strategy. Plus, they can establish latency, which is another fascinating aspect.
Latency.
It means the virus can go dormant inside the host's cells without causing immediate harm.
So it's like hiding out, waiting for the right moment.
Exactly. But under certain triggers, the virus can reactivate and cause disease. Some herpes viruses, like Epstein-Barr virus, are associated with certain cancers.
So they can be a ticking time bomb in a way. And their replication is also pretty complex, right?
Yeah, herpes viruses have large linear dsDNA genomes that circularize in the nucleus. They replicate using rolling circle replication, producing long chains of genomes that are cut into individual units. And their gene expression is tightly controlled, happening in stages like immediate early, delayed early, and late genes.
Mm-hmm.
They also bud through the nuclear membrane to acquire their envelope, which is pretty unusual.
That's a lot going on, so they're very good at persisting and even hiding within the host. Okay, that was quite a journey through DNA viruses. Let's switch gears to RNA viruses, which, as we mentioned earlier, all rely on RNA replicase.
Absolutely. And we'll start with positive-strand RNA viruses where the genome itself can act as messenger RNA.
So it's ready to be translated into proteins right away.
Exactly. Good examples here are MS2, poliovirus, and coronaviruses. MS2 is a small bacteriophage with a really tiny genome.
Okay.
But what's cool about it is that its RNA folds into specific 3D structures that regulate the translation of its genes.
So it's not just the sequence, it's the shape of the RNA that matters too.
Exactly. RNA can be both the genetic code and a regulatory molecule.
Multitasking RNA. And then there's poliovirus, which uses a different strategy to make its proteins.
Poliovirus is interesting because it produces one large polyprotein that is then cleaved into individual proteins.
So there's like a giant puzzle that's cut into pieces.
Yeah, a very efficient way to make multiple proteins from a single transcript. And its replication happens in the cytoplasm, using a viral protein called VPG as a primer.
Clever. Now, coronaviruses are also positive-strand RNA viruses, but they have these huge genomes.
That's right, they have the largest RNA genomes we know of. This allows them to encode a lot of proteins. Their replication involves first making the replicase, which then produces negative-strand RNA intermediates. These intermediates are templates for both new genomic RNA and smaller subgenomic mRNAs.
Subgenomic mRNAs.
These are smaller pieces of mRNA that are used to make specific viral proteins. And they assemble in the Golgi complex, which is different from many other RNA viruses.
So they have a more complex system for regulating gene expression.
Definitely.
Now let's move on to negative-strand RNA animal viruses. These have a genome that's complementary to mRNA, so they need to carry their own RNA replicase. Rabies virus and influenza virus are the examples here. Rabies has that distinctive bullet shape, right?
Yes, rabies virus, arabidovirus, has a bullet-shaped virion and a single-stranded RNA genome that is of the negative sense.
Negative sense.
This means it can't be directly translated into protein. So the virus packages its own RNA replicase to do the job.
It comes prepared.
Exactly. Once inside the cell, this replicase makes individual mRNAs from the negative sense RNA genome and also replicates the genome using a full-length positive strand as a template.
And all this happens in the cytoplasm.
Right, and new virions bud off from the cell membrane.
Okay, so it brings its own tools and doesn't need to mess with the nucleus. Now, influenza virus is also a negative-strand RNA virus, but it has a segmented genome and uses this cap-snatching mechanism.
Yeah, influenza is enveloped and its genome is split into segments of negative-sense RNA. It also brings its own replicase, but what's fascinating is how it makes its mRNAs.
Tell me more.
So its replicase has this ability to steal the cap structures from the host's mRNAs.
Steal, like literally.
Pretty much. These caps are essential for the host's mRNAs to be recognized and translated. Influenza just takes them and uses them to prime its own mRNA synthesis.
Talk about resourceful.
And unlike many RNA viruses, influenza replicates and transcribes its genome in the nucleus.
Oh, interesting.
And the segmented genome is key for antigenic shift. This is when two different influenza strains infect the same cell, and their segments get mixed up, creating new strains with potentially dangerous properties.
That's why we need new flu vaccines every year, right? Because the virus keeps changing.
Exactly. It's a constantly evolving target.
And the last group of RNA viruses we'll talk about are the double-stranded RNA viruses, with reoviruses as the example. They also carry their own RNA replicase, right?
Yes, reoviruses have a segmented dsRNA genome, and they keep it safe inside a protein shell called a nucleocapsid.
Safe from what?
From the host's defenses. Cells have ways to recognize and destroy double-stranded RNA, which is often a sign of viral infection.
Oh, right, the cell's own defense mechanisms.
So reoviruses do all their replication and transcription inside this nucleocapsid in the cytoplasm.
So it's like a little protected factory.
Absolutely. They make mRNAs from the negative strand of the dsRNA, which are then translated into proteins. And the genome replication is conservative, meaning they use the positive strands as templates to make new negative strands, forming new dsRNA segments.
So they've really got it all figured out in terms of protection and replication. Amazing. That covers RNA viruses pretty thoroughly, but we still have those fascinating subviral agents. Viroids and prions. Viroids are just RNA, right?
Yep, viroids are basically naked RNA molecules that infect plants.
No protein code or anything?
Nope. They get into plants through wounds and move around through plasmodesmata, those channels connecting plant cells.
Okay.
What's mind-blowing is that they don't encode any proteins at all.
So how do they do anything?
They rely entirely on the host's machinery. They use the host's RNA polymerase for replication, which happens through a rolling circle mechanism.
Like we saw earlier with some viruses.
Exactly. And many viroid RNAs can also self-cleave, which helps them replicate.
So they're like minimalist, parasitic RNA molecules.
Exactly. And they probably cause disease by messing with the plant's own regulatory RNA pathways.
So a tiny piece of RNA can wreak havoc. That's amazing. And then we have prions, which are even more bizarre infectious proteins.
Prions are truly mind-bending. They're infectious agents made entirely of protein, no DNA or RNA involved.
Wow. So how can a protein be infectious?
It's all about misfolding. Mammals have a normal prion protein called PRPC, but there's a misfolded version called PRPSC, that's the infectious agent. When PRPSC encounters PRPC, it forces it to misfold into the PRPSC form, like a domino effect.
So it's like a chain reaction of misfolding.
Exactly. This leads to a buildup of PRPSC, especially in the brain, causing those horrible neurodegenerative diseases like mad cow disease.
That's scary stuff.
Yeah, it really is. And what's even more fascinating is that we found prion-like proteins in fungi and even humans that aren't harmful and might even be beneficial.
So this misfolding phenomenon might have other roles besides just causing disease.
That's what researchers are trying to figure out. It really challenges our understanding of what an infectious agent can be.
And lastly, the chapter includes a glossary of terms, which is always helpful when you're dealing with so much technical jargon.
Absolutely. Terms like concatemer, negative and positive strand, replicative form, rolling circle replication, viroid, and prion are key to understanding this field.
Well, that was an incredible deep dive into the amazing world of viral genomics. We've covered so much ground, from the basics of their genomes to these mind-blowing examples of prions and viroids.
And hopefully, our listeners now have a solid grasp of how diverse and ingenious viruses are, how they replicate, and how we classify them. We touch on everything from the structure of their genomes to the intricate mechanisms they use to take over their hosts.
And with that, I think we've successfully completed our mission of summarizing the entire chapter, covering all the major points, theories, findings, and examples from this chapter summary on viral genomics and diversity.
Absolutely, we've covered it all.
Thanks for joining us on this deep dive. Until next time.